Okay, so my topic was investigating autophagy pathways in retinoblastoma. And so my name's Coven, and I'm a current 11th grade student at DeBakey High School. Okay, so essentially what is retinoblastoma, right? It is a eye tumor present in the retina, and it's primarily present in young children, so children under the age of 5. So as you can see in the diagram that I have, you have a healthy eye, and then you have a retinoblastoma where you can see the tumor. I don't know if you guys can see my cursor, but there's a tumor present in the back of the retina. And essentially what my research focused on was delving into the cellular process of autophagy, which is kind of like this cellular cleanup and cells recycling old material, um, degrading, it's just essential type of cleanup for cellular components. And so it can actually play a dual role in cancer development. It can offer both protective and harmful effects, but essentially what my research aims to do is see how that can actually benefit retinoblastoma patients by examining their pathophysiology and potentially creating a way for new therapeutic strategies. And so this can help in early detection, diminishing risk, and helping treatment complications. And so some of the literature review that I conducted, so there was a paper that I found from Unit All. This is kind of a little bit old in 2011, but it did a really good job of explaining actually how retinoblastoma works. Um, Especially in the molecular level through RB1 gene mutations, and then kind of explaining how not only internal but also external factors can actually help in the development of this cancer, and as well as explaining different current therapeutic options. And then there's another paper that I found by Wan et al. 2021, which dived into telling the cellular mechanisms of retinoblastoma, kind of how autophagy can help self-degradation, recycling old materials, and how, like I explained, how that actually plays a dual role when it can actually be both positive and negative. And then what the key proteins and genes were that were involved in that process, which helps to provide a more targeted approach. And then the other main research paper that I found was from Gu et al. 2021, which really just explained the research gaps in current retinoblastoma research. And so I used that to actually figure out, okay, what can I actually do and what can I focus my research on? And so we needed a more advanced and precise technique to actually target certain molecules and genes. And so the methodology and approach that I used. So essentially what I did was I used bioinformatics, or what I was planning on using is bioinformatics analysis. So utilizing both genomic and proteomic data, so data about proteins and genes. And then I took that from data sources from different public gene libraries, data such as RNA sequencing data, and then using computational tools to actually be able to investigate these pathways and figure out, okay, what genes are actually responsible. And so what I was able to find was there were differences in the autophagy-related gene expression in retinoblastoma tissues. So figuring out, okay, between healthy and cancerous tissues, what exactly are the key genetic differences? And then I use that to actually figure out, okay, how can these genes actually influence the tumor behavior, things like growth, creation of a tumor. And so just basically understanding the underlying pathology of retinoblastoma at a molecular level. And essentially what this can do, it creates more targeted therapies and provides a more holistic approach as to how we can go about treating retinoblastoma through therapeutic interventions. And so in conclusion, some of the contributions and results that I found was an insight into kind of the autophagy related mechanisms. So actually how these autophagy pathways can contribute to retinoblastoma, which provides an understanding of how retinoblastoma works, how our actual like body and cellular processes can contribute to retinoblastoma. And I was able to identify molecular targets, which helps in creating potential genes and pathways which we can actually use to create drugs in the future that are targeted to those genes that are responsible for retinoblastoma. And some of the limitations that I found was there's a limitation in the range of the data being analyzed. Retinoblastoma is not that popular. It's quite rare in some form. But so I found a big limitation in the amount of data that was available and also the scope of the data, right? And then obviously in the future, we need to have a more clinical application of my findings or all these findings in general of the autophagy pathways and being able to implement them to clinical trials to actually test out whether these identified, whether through humans or mice or some other biological model, being able to test whether these genes can actually suppress retinoblastoma. Because right now we have no way of actually knowing, oh yeah, this gene is actually responsible. This pathway is actually responsible for retinoblastoma. We'd have to actually test it in a lab. And so then I want to share my research experience. So Professor Virgil was really helpful in teaching me how to actually turn my research idea into an actionable like paper and actually teach me how to write and how to think. And Dr. Prahlad's guidance was really helpful in understanding the kind of scientific aspect behind this, how to actually conduct research, how to write my paper in a way where it's easy to understand for people in a way that encompasses all my ideas that I need to be able to express. And in general, this was highly just entertaining and informative. It really sparked a passion for research that I didn't know I had. And then I was able to I found out that I actually really enjoyed reading scientific papers. Like I thought it would be boring at first, but then through this extensive reading and doing all my literature review and actually writing this research paper, I actually found that I really enjoyed this research process from start to finish. And yeah, that's it.
Hey there, thanks for sharing your thoughts and the depth at which you've gone into the research. And by the way, Kavin and his sister, they are all part of our research program. And this is your second one, right, Kavin?
Yeah, this is my second one.
Fantastic. You want to just turn off the presentation and share your thoughts about the research program and how it's helped you overall?
Yeah, so this research program, as well as also my last one, has really helped me delve deep into like things that I'm passionate about. So I originally I didn't know that I enjoyed research or things like that. But then through actually doing it, I found that, oh, there's actually a way for me to contribute to the scientific field, even as a high schooler, which I found like really cool. As like I can actually make an actionable difference through doing something that I actually enjoy doing, through delving into like my scientific passions. And just it's really I found it like the guidance especially really helpful. Like, oh, I'm not just all on my own here. I actually have some guidance, I have help, so I know what I'm doing. And to make something that's actually like high quality, like a high quality research paper, I found that like really cool and really interesting.
Yeah, yeah. Wonderful, Kavin. Great job.
Right.
A big round of applause for Kavin.